This
And from
conference will
one point,
now be recorded.
hello. So this is about the normal, the fluorescence, that is a spontaneous emission, is getting amplified in the presence of stimulated emission. So because of the gain that is available in the structure, this stimulated emission is getting narrowed. This is the absorption spectrum of the dopant. This is a normal emission of the dopant, and you can see this is the amplified spontaneous emission. It is much narrower than the normal fluorescence, but it is an amplified version of the fluorescence, the normal spontaneous emission. So even without um, expecting any lacing from this microcavity, so this is the microcavity that is uh, kept inside this chamber, and you are exciting this with a green laser, and the emission from this microcavity is given to a spectrometer. And what is happening here is you can uh, have a tuning of the emission wavelengths simply by changing the diameter of this structure, this hollow structure. That is, this is a I mean lower diameter, and these are the higher diameters. So when you have higher diameters, the shift in wavelength is very much visible. So what is happening here is there is reabsorption, absorption and emission, and the reabsorption of the emitted radiation that is happening because of the stock shift. So you have fluorescent material inside the gain medium, inside the microstructure. It is excited with a green laser, and after absorbing the light, it emits the light, and that emission is being amplified. That is coming out as the amplified spontaneous emission. But the lower portion of each of this curve is reabsorbed by the same material. So you can see there is a spectral overlap, that is absorption and emission, they are overlapping. So because of this overlap, the lower wavelength region will be reabsorbed by the same material. So there is a chance of stock shift, and the emission wavelength will be shifted towards the right. That means when the size is increased, so if I increase the diameter of this microcavity, it actually leads to a more reabsorption. That is, the length is actually increased. The interaction length of the light matter is actually effectively increasing. So that enhances the stock shift, and that results in the shifting of the wavelengths towards the no, longer wavelength side. And here also the second type of tuning is the temperature tuning. You can apply temperature within this chamber, and then you can see what kind of changes in the emission spectrum that is happening. So in this way also you can tune the output emission wavelength, because the applying temperature, you can expect a change in the diameter or an effective change in the refractive index. So whichever is dominant, depending on that mechanism, you can also uh, expect a the wavelength. So two types of tuning is possible from this kind of material. And another one is, instead of heating by externally applying any heat energy, we are making use of two lasers. So in order to heat the sample, this is the fiber, that is the microcavity, doped microcavity, and we make use of a continuous wave laser. So in the ultraviolet region, that is 403 nanometer, that is focused onto the fiber, and this fiber is being pumped with the 532 nanometer green laser, and simultaneously these two radiations are falling on the fiber. So because of the pumping, there is lacing, and one particular frequency can be monitored while it is being radiated with the second laser. and we can also find what is the effect of this second laser on the expansion of the fiber or the change in the refractive index, and in that way you can also so something like an all optical, like you make use of two lasers without making use of any external agency to change the temperature of, because this will be more effective in remote regions wherein you have you need to access the things only using an external radiation. So this is a work that was done not in our lab, but in collaboration with our laboratory. It was done in UK. So this is a silica gel coated spherical microresonator. So in this figure you have a chamber, and this can be evacuated, and then necessary ammonia gas can be applied, and on a substrate you have kept a silica gel coated spherical microresonator. This is a microresonator, spherical in shape. This has ultra high sensitivity and the ability to detect ammonia gas concentration in the air. So so it detects the ammonia vapor concentration in air at a constant relative humidity. So the excitation is by evanescent coupling. So you have a superluminescent diode. You can couple light from this diode, this source, to the optical fiber, and you have a tapering of the fiber, as we mentioned earlier, so that from the fiber taper the light can effectively come out and get coupled to this microsphere, and the waste diameter is as small as the beam, I mean the fiber waste diameter, because fiber is tapered, and after tapering its diameter actually reduces to around 3 to 4 micrometer, where it is in contact with the sphere, microsphere, and you can continuously monitor the output spectrum that is available in the optical spectrum analyzer in presence of ammonia gas. So the sensitivity obtained for this particular case was 34.78 picometer per ppm. So this was the sensitivity obtained for this spherical micro cavity. One interesting result that we obtained was something like the whispering gallery modes available in a graded index structure. This is very interesting, because in this particular result what we observed was that the selected frequencies in a microcavity can be changed, by simply changing the length of the fiber. Okay, so because of the graded index structure available, so you have a continuous variation of refractive index in this region. So it not only contains the gain media, but also the additional material for refractive index variation. So once we excite this fiber from transversely or from the side using a green laser, it generates the whispering gallery mode, but the generated modes, they propagate through the fiber, and while propagation they are getting focused because of the refractive index variation available in. So continuously the circumference is actually, that is the mode volume, the volume of the electromagnetic energy, so that is actually continuously getting reduced. So accordingly the emission spectrum is continuously varied. If this is the emission spectrum at a particular location of the fiber, suppose I am taking this much length of the fiber, we are talking about millimeters and centimeters obviously, but if I am taking this much length of the fiber, I have the circumference of this much, which leads to a set of frequencies, and if I am choosing this much length of the fiber, I am obviously taking a different set of frequencies. So this can be continuously changed, and after certain distance we see that the lasing actually dies out because of the losses, but within this region you can see there is huge variations in the selected frequencies, and also the FWHM correspondingly, we know the circumference will determine this frequency separation. So the frequency separation will be different if you have a length of this much. The frequency separation will be different if I am taking this much length. So that is obvious from this spectrum. So you have different spectra, and their locations are different, their frequency values are different, their FWHMs are also different, and this may find huge applications in distributed kind of sensing, wherein you make use of the same fiber and you want to measure the parameters, various physical parameters from different locations using the same fiber. And one important thing to be noticed here is that the laser emission in all these cases, they happens in all the 360-degree. They are not at all directional. So you have a microcavity, something like this, in the form of a hollow cylinder, like in the case of a fiber doped with a gain medium. So you pump it from the side, and the laser actually oscillates, and the emission is happening from all the 360 degrees. It is not at all directional unless it is taken from this side, because in WGM it is actually oscillating in this way, and it is more fields are available from the sides of the fiber compared to the end. So usually people are collecting the WGM output from the sides, from all the 360-degree from the fiber. So due to the same reason, if you want to have a considerable energy in a particular direction, it becomes very difficult to have a useful energy in a particular direction. So one solution for this kind of in order to get better directionality, what we can say is that what we can do is you make an asymmetric cavity. This is something like a symmetric cavity. What happens if the cross section is something like this? That means you have a smaller thickness of the film over here and a larger thickness over here. So once this is pumped and light is WGM starts oscillating, then you can see that there are more chances that more leakage from here. So from the simple ray picture and the condition for total internal reflection, you can show that the more light will be coming from this direction, in this direction, compared to this direction or any other direction. So because of this particular structure, so such a laser was tested, and this is the figure of that laser while lasing. So it was pumped using a grain laser, and this is actually lasing. The photograph shows the lasing of the asymmetric cavity. So you can know that very well, see that this region is much thicker than this region. So we can expect more emission from this region. So that experiment was conducted, and we have demonstrated that a asymmetric resonant cavity, asymmetric ring cavity, will be more directional than the symmetric ring cavity. So one problem we have already discussed is about the directionality of emission. The other one is the photostability of the materials that is doped in the fiber. So the fiber once doped, the material once doped inside the fiber, after getting exposed to the laser continuously for a period of time, so there is a process of degradation that is happening to the fluorescent material. So that is actually called the photo bleaching. Okay, so we say that if the photostability is poor, so the material will degrade very easily. So it is very interesting to have a better photostability for all the samples that is fabricated. So this is a study conducted in terms of the photostability, and we see that this is a comparison between a step index, a hollow, and a graded index fiber, and they were continuously exposed to the laser pulses, and their emission was noticed, and it was found that the rhodamin 6G based for rhodamin 6G based microcavities or fibers, the graded index structure actually uh, offers a better stability compared to the other two structures. And one method that we tried was to incorporate some metal nanoparticles into these structures along with the fluorescent material. So along with the fluorescent materials, uh, silver nanoparticles were also incorporated, and that resulted in better photostability, because we know there is a uh, phenomenon called SPR, surface plasmon resonance. So once that happens, the radiative decay will increase the field around the Ag nanoparticles. The silver nanoparticles will be much larger. So the local field enhancement happens around the silver nanoparticles, and whatever fluorescent molecules sitting very close to such nanoparticles will be more efficiently excited. And in a way, it is an indirect excitation, not a direct excitation. So in that way, the nanoparticles can shield the organic fluorescent material, as well as it can offer a better and more efficient excitation mechanism for them to have a better emission and better photostability. So it is proved that the incorporation of metal nanoparticles can reduce the photo bleaching. That means it can enhance the photo stability, and that studies have been conducted, and similar studies were done with silver nano wires as well, and for a particular concentration of such silver nanowire, we got a very low threshold for lasing compared to the microstructures that doesn't incorporate any silver nanoparticle. And other things, we are very much familiar with the erbium doped fiber amplifiers used in communication. So wherein the silica fiber is doped with erbium, and it is pumped with a 980 nanometer laser, and we are expecting this lasing transition to happen at 1550, so that the signal at 1550 can be amplified. And this is the experimental setup for erbium doped fiber amplifiers. You have 1550 signal coming in, and 980 nanometer, a local pump laser diode is incorporated here using a coupler, and this is the region over which you are having this erbium doped fiber, which actually replaces the original fiber. So it can amplify the 1550 nanometer signal because of the exclusively the property of the erbium doped erbium that is doped in the structure. A similar thing can be imagined in the case of a plastic optical fiber which is doped with a fluorescent dye, but the only difference is that you can have a visible radiation that is passing through this fiber, and it is simultaneously pumped, as well as a signal is coupled into the fiber. So a signal as well as a pump, they were coupled to the fiber in space and time. They are coupled together and then applied to a piece of dye-doped polymer fiber, and then we studied the emission or the amplification behavior of this fiber for that 95 nanometer signal that is given from a optical parametric oscillator. And this is a signal and this is the pump, and both are combined in space and time and given to the dye-doped So we expect huge narrowing from 42 nanometer to 7 nanometer width for the emission, and also about 18 dB. So this is a work done in 2007 by one of the research scholar in the USAT. So the gain of 18 dB was observed for a fiber of length 8 centimeter. So the length of the fiber was simply 8 centimeter, and this much gain was observed. And one important point to be noticed is that the gain spectrum is very wide. So it's not only the 595 nanometer that can be amplified. You can choose a range of wavelengths which can be amplified because of this broad gain spectrum. And what we have seen earlier is that the micro ring embedded structure can result in a single mode operation of the WGM lacing, but the power, useful power, that can be coupled out of such devices is very, very feeble. And one possible mechanism is to amplify that single mode using an amplifier. And we are currently conducting such an experiment wherein you have an oscillator. Oscillator will be a hollow fiber, that is a micro ring embedded fiber. That is, this is something like a hollow structure, but inner wall of this is doped with a gain medium. So so that is acting as an oscillator, and then this is coupled to an amplifier. So amplifier can be a graded index fiber. So both this oscillator will look like this. That is, it is going to be a micro ring embedded hollow structure which generates only a single mode, and this is a amplifier is going to be a graded index fiber, and this single mode is coupled to this amplifier so that it can just amplify that single mode. This is schematically represented here, but this is a multi-mode that is shown, but it can very well amplify the multi-mode as well, or even if this is a single mode that is given in, so the amplifier can amplify that single frequency. That is the concept. But the major hurdle that we face today is to couple light, which is already very feeble, from this oscillator to the amplifier. So air coupling uh, or the surface coupling from oscillator to the amplifier is very tedious, and nothing is getting coupled. So what we currently doing is something like a mechanism like this. So on the same piece of polymer, we are actually fabricating a micro ring embedded hollow fiber, and a part of this will be a graded index structure. And once this is drawn in an optical tower, you have a combination of these two, that is micro ring embedded structure and the graded index structure available on the same piece of fiber. So that is the oscillator and amplifier in the same piece of fiber. So currently we are working on that, and even before that we have just tried this kind of amplification using this is from spectrum from the micro ring embedded structure, and this is a spectrum of the graded index, and this amplification is currently being studied. That is around 24 dB gain we reached, but the coupling is still very poor, and we are working on this single unit. So I am coming to the conclusion of the first portion. The doped polymer fiber can be very good candidate for the fabrication of optical microcavities. Polymer-based microcavities can offer tunable low threshold lacing in the visible region. Graded index, hollow, micro ring embedded types of polymer microlaces can be used for sensing a variety of physical parameters. Photostability of organic dye-doped polymer laces can be improved by the incorporation of metal nanoparticles. Micro ring embedded hollow Micro cavity offers strong modulation leading to single mode operation. Improved directionality of the WGM lacing can be realized by an asymmetric cavity. So I would like to acknowledge my students and my funding agencies. I acknowledge the funding from DST, SERB, as well as the collaborations that we have between these two universities, and our students were they had the privilege to work in these universities collaboratively for many of the results that we obtained. And Rupa Venkataraj, she recently graduated, and Jason and Linslal also, they graduated last year. And currently Anand and Arindam Sarkar are working in the same problem. Thank you very much. So now I will move to the next topic, which I will finish as early as possible. Hello, is it visible? Hello?
Yes, sir. It is visible.
The next slide is visible, right? Okay.
Yeah.
Okay. 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 So now I will just give an overview of the different types of optical sensors, but mainly fiber-based sensors. These optical sensors are used for contactless detection, counting, positioning, etc. These are the major applications that we see in our day-to-day life in commercial sensors. So it can be a through beam sensor, a retroreflective type, or a diffuse reflection type of sensors. That these are the things that is more common in our day-to-day applications. And optical sensors are both sensors for light and sensors using light for sensing non-optical properties such as forces, displacements, tilts, temperature, velocity, pressure, chemicals, and electrical properties, a variety of parameters that can be sensed using or optically. So these are some of the day-to-day applications that we encounter: the ambient light sensing, the optical heart rate sensor, then breath analyzing, then the water level sensor, etc. are simple sensors that we use in devices. But when we come to the fiber optic sensors, we have an optical fiber which interacts with the measurement, and this measurement can somehow alter the characteristic of the light, some characteristic of the light that is passing through the fiber. It can be phase, it can be intensity, or it can be the wavelength of the light that is passing through the fiber. So it can be altered. So the fiber optic sensors, it uses the light guided within an optical fiber to detect any external physical, chemical, or biomedical parameters. So this is a broad classification in terms of what parameter or what characteristic of the light is being altered while interacting with the external medium. It is classified as intensity based sensors, then phase, then the spectrum. So either the intensity can be modified, or the phase can be modified, or the wavelength can be modified. Other one is the extrinsic and intrinsic. This classification means, in the case of extrinsic sensor, in the just like the thing that we were discussing in the last slide, most of the common applications, the light has to come out of the fiber, then interact with the external medium, and then get altered some of its characteristics, and then it will be detected later. But in the case of an intrinsic kind of fiber optic sensor, the light will be maintained within the fiber. The light will be guided through the fiber. It is not coming out of the fiber while interacting. So while the light is within the fiber, it interacts with the external parameter. It can be displacement, force, pressure, temperature, anything. So the signal developed by the detector is very important in any optical fiber sensors, and the signal developed by the detector depends on many factors. I've just read out the power emitted by the source, the coupling efficiency of the input and output fibers with the modulating element, then the transmission efficiency of the fiber, the response of the modulating element, then the responsivity of the detector. So all these will determine the signal developed by the detector. And coming to the most simple type of a fiber optic sensor, it can be an intensity modulation. So we need not worry about what happens to the spectrum, or what happens to the phase of the light that is propagating through the fiber. So it will be simply coupling, like some lights, something like a different axial or lateral or angular displacement that is happening between the fiber 1 and the fiber 2. It can be lateral, angular, or transverse. So this kind of variations can simply develop a signal in the output. But here you see there is a combination, say single fiber and a pair of fibers. So which can act as a differential sensing, differential fiber optic sensor. So depending on the alignment, the relative positions of the fiber 1, the transmitting fiber with the receiving fiber 1 and receiving fiber 2, you have different values of output. It's a very simple one to understand. And in many of the other type of fibers, we make use of the evanescent waves. That is the light that is coming out, the energy that is coming out of the fiber, that is leaked out of the fiber, so which interacts with the measurement and thereby alters the intensity or some characteristic of the light that is passing. So in order to understand, I will just go through the waveguiding condition that is happening in any waveguide. So this is a simple planar structure. You can imagine a cylindrical waveguide also. So the basic condition for the light to be contained or confined continuously within this structure is, within the core, they have to replicate, like constructively interfere. So after this reflection, it gets reflected from here and it continues in the same direction. So while continuing in the same direction as the original wave, the wavefronts overlap. So their wavefronts, if you imagine this as a plane wave, these wavefronts are plane. So the plane wavefronts overlap even after many such reflections. Also, this is a condition for waveguiding. For all other angles, what happens is that the destructive interference of the light happens, and those angles are specifically not permitted for transmission. So each of these angles actually gives you a mode or a specific electric field pattern within the structure that is supported within the waveguide. So you can have many such angles if the waveguide is a multi-mode one. And if you consider two such rays propagating, uh, you can have multiple parallel rays, but for simplicity we consider two such rays propagating in the core, and they have to get added at different locations. Their electric field values will be different at different locations, and if you calculate that electric field values across this structure, we can find that it is a harmonically varying function along the y direction. Along the y direction, it doesn't have any time dependence. It is a standing wave pattern, the first term, and the second term is a traveling wave. It is a time dependent term. So a harmonically varying electric field pattern along this y direction is propagating along the z direction in a optical fiber or any waveguide. So each of these directions actually leads to one such mode. So this is a mode, this is another mode, and this is another mode. So you have different directions. So in the ray picture, we depict them like this rays. In the wave picture, we depict them like this modes. Okay, so but each of them are originated from a particular angle of incidence, and all angle of incidence are not permitted. That is an important thing. But one important thing to be noticed that if each of them are modes, some of them are having very less number of reflections, the other ones are having a larger number of reflections. Whenever you have larger number of reflections, those are corresponding to the modes that are higher order. These are higher order modes. They have more number of reflections, and lower order modes are less number of reflections, and this corresponds to the lowest order mode, and this is a higher order mode. And one thing to be noticed here is that the penetration into the cladding, as we mentioned earlier, there can be a tunneling. This is a kind of optical tunneling. So in fact, this is a forbidden region for the photons concerned because of the lower refractive index, but they tunnel into the cladding. So the tunneling is more for the higher order modes. So this is a higher order mode. So obviously its mode, that is the electric field pattern, is actually leaking more into the cladding. For a lower order mode, it is leaking very shorter distance into the cladding. So what is leaked into this cladding is actually termed as a evanescent field. So you can have evanescent field in all these kind of waveguiding structures, even though you see that some energy is actually leaked out. It can be useful for many applications, including sensors. So you can effectively calculate the diameter of the mode, that is a field diameter. So the field diameter in such a case, for a single mode case, for example, is not the diameter of the fiber, but it actually extends into the cladding, and this extended region is the field corresponding to the evanescent wave. You can see there is an exponential decay into the cladding. So it actually follows this relation, the intensity from this point to this point or any other point away from this interface follows this relation. Okay, so once you have this kind of evanescent wave present in your guiding structure, so that can be easily exposed to the external medium by removing cladding from certain region. For example, this is dp, that is the penetration depth, the depth to which the evanescent field is penetrating into the cladding. That is called the penetration depth. So if you remove the cladding, what happens? This field has an opportunity to interact directly with the external medium. So that is actually what is shown here. So this is an optical fiber with core and cladding, and for certain region, the cladding is removed. For a length L, the cladding is removed. So the field, evanescent field, is allowed to interact more easily with the external medium. For example, a sensing region. So this sensing region can be a solution or a chemical which can directly interact with the field. And one interesting relation, that is the field that is propagating from here to here, that is passing through this uncladded region, follows this relation. So the power at any particular length will be proportional to power at zero distance from here times e raised to minus gamma L. That is the evanescent absorption coefficient is this gamma, and this gamma is nothing but R times alpha, where R is the proportion of power that is guided through the core, contained in the cladding. So the proportion of power in the cladding is determined in terms of the V parameter, So you may be familiar with the V parameter, but V parameter is the characteristic value of a fiber or any waveguide, which will be determined by the geometry as well as the N1 and N2 values and the wavelength of operation. So once you know this R, and once you know this alpha, so this R is the proportion of power that is in the cladding divided by the total power, and this alpha is the bulk absorption coefficient, the absorption coefficient of the medium. So suppose you are immersing this uncladded region into a liquid, so that has a absorption coefficient, that value is this alpha. So it follows this relation. So by allowing this external medium to directly interact with the power that is being propagated through the fiber, it is a very good method to sense different chemical species over here. So these are some of the examples of evanescent wave fiber optic sensors. So for example, this one doesn't make use of any tapering of the fiber. This is the optical fiber, and this region is uncladded. And this entire thing is inside a cavity, and this is having a coating. This uncladded region is given a coating. As we mentioned earlier, it can be some certain functionalization can be done over this core region, so that it can attract some other biomolecules through binding, and in that way it can change its surrounding refractive index, so that that changes can be monitored by a spectrometer. But here you can see there is a sol gel coating given to a tapered fiber. The fiber is actually a tapered one. In this region it is tapered. The tapering allows the more field to leak out. More evanescent field is exposed to the external medium by tapering. So it can interact with the sol gel, and the sol gel is something that may be interacting with the external gas or whatever parameter. So in that way, here also the evanescent field is interacting with the external medium. Here, a tapered fiber optic sensor, as we mentioned earlier, the tapering is a very good sensing region. Not only we have to unclad the fiber, but also make it a tapered region. Here is a tapered tip. This tipping, this kind of tapered tip is also found to be very good region where the field can interact more efficiently with the external medium. So evanescent coupled nanowire, for example, over the over the fiber which is tapered, this is a sensing region, and a nanowire is actually formed by different techniques are there for coating this kind of nanowires or nano cylinders, whatever. A nanowire is sitting over this sensing region. And this kind of sensing we have already discussed in the first session. So you have a fiber that contains the light, and this is the in and this is the out, and in between it is tapered, and in the tapered region a micro cavity is placed. So some frequencies can be coupled into the microsphere or micro cavity or micro cylinder, whatever shape. So the output frequency analysis or the spectrometer analysis of the output will show that certain frequencies are missing. So in that way also the evanescent field plays a huge role in all kinds of fiber sensors. Another important class of sensor is based on the surface plasmon resonance. Surface plasmons are the collective oscillations of electrons. When excited with light under certain specific conditions, under some conditions, we can excite this surface plasma wave to oscillate in between, at the interface between the metal and a dielectric. For example, this is a Kretschmann configuration to generate such surface plasmons. This is a prism, and this is a light source, and this is the detector, and you can continuously change the angle of incidence, and you can continuously monitor your output spectrum from here. And you see that at a particular angle, these surface plasmons are generated. They are excited, and it is generated at this metal dielectric interface, and this happens, and this field, that means the evanescent, a huge evanescent field is generated during that process, and this field, evanescent field, actually extends into around 50 to 100 nanometers, and during that process it can interact with any external medium that is present here. It can be chemical or biological kept over here, and you can and this angle at which this surface plasmon generation happens effectively depends on the dielectric constant. So obviously what is present here will determine this angle. Continuously monitor the output spectrum, you will find a dip at a particular angle of incidence, and you allow the chemical or biological change to happen over here. So that will change effectively the dielectric constant of this medium, and that is going to be reflected in at a different angle, so from which you will be able to understand the changes here. One example shown here. So this is an inverted structure, the same structure, but only an inverted version. So light source is falling on the, coupled through a prism, and this is a gold layer, a thin layer of gold, and over which you have antibodies immobilized, which is through functionalization you can immobilize certain antibodies, and these are the antigens, and how much antigens are getting binded to these antibodies can be directly monitored by changing this angle and then seeing how much shift in this dip is occurring. So that is a huge area of surface plasmon resonance, and a lot of work are being done in this area. And one broad classification of optical fibers is something like a Mach-Zehnder interferometer kind of thing and a Zagnac interferometer. In a Mach-Zehnder interferometer, a collimated beam is passed through a fiber optic coupler, which splits the beam into two equal amplitudes. This is a coupler and this is a source, and it splits the beam into two equal amplitudes. The two beams are then passed through a second mirror through the two arms. So usually one of the arms is called a sensing arm, the other one is going to be a reference arm, and reference arm will be completely completely covered from the external perturbations, isolated from the external perturbations, but from here it is open to interact with the external parameters. So whatever the phase difference happening, and that they will be combined, and that will be monitored through these two detectors, two intensities I1 and I2, or the voltages can give the idea of the kind of phase change that is going on in the one of the amps. So that is a Mach-Zehnder kind of interferometer. This is the corresponding bulk setup. If you set up such a experimental setup in the lab on a table, it will look like this. It will have several mirrors and beam splitters, laser source and detectors and screen, etc. But it is very simple setup to replicate this system using two optical fibers and two couplers. Here there is a Sagnac interferometer. Sagnac interferometer is usually used for orientation sensing or rotation sensing. So you can see the bulk setup. It contains a beam splitter and three mirrors, so that the light given from the source is allowed to travel in two opposite directions. So they circle in this direction clockwise and in the anti-clockwise direction, and the entire setup will be rotated in any one of the direction, so that there will be a time difference for the light to reach the detector in the clockwise direction and the anti-clockwise direction. From that time difference, you can translate into a phase difference, and that will be used for sensing. But again, if this is a bulk setup which makes use of a 3 mirrors and a beam splitter on the table, this can be very simply replaced with a coil of optical fiber and a coupler, something like this, and a source and a detector, and it can be miniaturized, and it can be applied for many applications, including space applications. So usually a phase modulated optical fiber sensor is something like a Mach Zehnder interferometer only. This is this kind of setup only. Okay, so wherein you have a reference arm, a measurement, this is a sensing arm, and this is completely isolated from the external perturbation, and you can calculate the phase difference happening by beta times L. So beta is 2 pi by lambda times n effective. So I'm not going details into the theory, but the thing is that there is a phase difference that is generated because of the interaction of this part of the fiber with the measurement, and that can be very well monitored by measuring the outputs I1 and I2. So in that way, you can have it can be a change in length, or it can be a change in refractive index that causes a change in the phi, that is the phase. You can translate this into intensity also. So the intensity of the first detector, reaching the first detector and the second detector, they follow this relation, and you can have if delta phi is equal to 0, the entire power will be in one of the amps, and the other will be 0, or vice versa, depending on the delta phi value, or it can be equally divided or divided into I1 and I2. In all case, I0 is equal to I1 plus I2. So in the case of rotation sensor, as we mentioned earlier, the Sarnack effect is made use of here. That means you have a coil of fiber, and the light being coupled into both the ends simultaneously. They are allowed to propagate clockwise and counterclockwise, and if the entire system is moving or rotating, it can clearly sense that rotation, because there will be a phase difference between two counter propagating waves because of the rotation or the movement of the entire system. So again, this is a highly miniature version of this tabletop Sagnac interferometer. So you can calculate that delta t, that is difference in time for the counter clockwise propagation and the clockwise propagation, which can be translated into delta phi, and you find that this delta phi can be considerably large when you have a large number of such fiber turns available. You increase the fiber turns like 1,000, 10,000 turns, so that even a slight orientational difference or slight movement or a slight rotation of the entire system, entire platform is measurable. This is a very good example shown here. If you attach such a system to the hour hand of a clock, okay, the hour hand of a clock, its rotation rate is just 30 degree divided by 3600 second, that is roughly 1.46 radians per second. So if it is a single turn, such kind of system will generate only a phase difference of 7 into 10 raised to minus 7 radian. Okay, but if we simply increase the number of turns of the fiber in such a system, okay, so it can make huge difference. So making 10,000 turns, this time difference between the two counter propagating beams can be larger, and it translates into a larger value of delta phi. So it can be 7.2 milliradians. From this very small value, it can reach 7.2 milliradians by increasing the number of turns. And another class is fiber Bragg grating. So fiber Bragg grating is it is something similar to a one-dimensional photonic crystal, like you have periodic variations in the refractive index written onto the fiber core by making use of UV radiation. You make interference patterns focused into the core of the fiber. This fiber obviously is a photosensitive fiber. It contains a large amount of GeO2, that is germanium oxide, which is a photosensitive material. So in the core of the fiber, you can write these kind of gratings, which can act as an optical filter. So depending on the periodicity of the grating that is written, this is the periodicity, the distance between any two these refractive index variations. So if it matches this condition, if it obeys this condition, lambda is equal to 2n effective capital lambda. This capital lambda is the periodicity. This particular lambda of the light will be reflected back. Okay, so that is the condition to be satisfied. So this is the incoming radiation and some part is reflected back. This is a reflection spectrum, and the rest of the thing will be transmitted, and in the transmission spectrum you will find a dip. So in a fiber Bragg grating, the condition to be satisfied for coupling of power with a forward propagating and a backward propagating mode is that this difference in beta values of these forward and backward propagating beams should obey this condition, that is beta minus beta is equal to capital K, where capital K is 2 pi by capital lambda. This capital lambda is the periodicity. Okay, so from these values we can see that the lambda b, that is the wavelength that is going to be reflected back from such a device, is going to be very small if you are considering, or I mean, if you are considering the communication wavelength somewhere around 50 nanometer. If you want to fabricate such a fiber Bragg grating, the periodicity requirement is going to be very stringent. From this equation, you put 50 nanometer here and plug in values of n effective, you can see that this capital lambda is going to be something like a few hundreds of nanometers, something like 500 nanometer. So the distance between these two high refractive index regions written in the fiber core has to be around 500 nanometer for a fiber Bragg grating. So that is a very expensive business, and that is the idea behind this optical filtering using fiber Bragg grating. So coming to some of the characteristic features of the fiber Bragg grating, it is a reflective type filter. It is not like other type of filters. The demanded wavelength is reflected instead of transmitted. It is very stable, and after annealing, so after writing the grating using UV radiation, it has to be annealed. The annealing makes it makes the changes in the refractive index permanent, and the reflective spectrum is very stable over a long period of time. It is transparent through the wavelength signals. The gratings in fiber are gratings are in the fiber and do not degrade. It will not degrade soon, and whatever written on the fiber after annealing is almost permanent. It is an in-fiber component and easily integrates into other optical. It is not an external component. Obviously, it can be written onto the fiber which is used for communication. So it can be very well integrated easily. So these are some of the equations used for strain sensing, temperature sensing, and pressure sensing using such fiber Bragg gratings. Obviously, the capital lambda, which is the periodicity that is written onto the fiber, is going to change when we apply the strain, when we apply the temperature, and we apply the pressure. So depending on these parameters, the sensitivity of such sensing devices can be calculated. For example, in the case of this pressure, this is d capital lambda by dp. So the pressure is going to change your periodicity. And what is distributed sensing? So once a fiber Bragg grating is used for a sensing, can it be used the same fiber? Can the same fiber be used for sensing the same parameter at a different location? The answer is yes, because you take the same fiber and make different gratings at different locations. This one corresponds to, this periodicity corresponds to the reflection of lambda 1, lambda 2, lambda 3. So the periodicity varies at different locations, and at various locations you have written the fiber Bragg gratings, and it can be installed in huge structures and bridges and railway tracks, etc. And you send a broadband light source for exciting this fiber, and you can monitor each of these wavelengths in the control room, and you can easily predict from which location this particular lambda is arriving, or what kind of change of strain, temperature, everything is locally happening at that location. So another class of long fiber grating is a long period grating. So we have seen that in a fiber Bragg grating, there is a coupling happening between a forward propagating mode and a backward propagating mode. Okay, but here the coupling actually happens from a mode, a core mode to a cladding mode. A propagating mode in the core couples some of its energy into any one of the cladding mode. So this is the condition. This is for the cladding mode, and this is the uh, um, core mode. This is for the core mode. So this is the beta value of the core mode, beta value of the cladding mode. The difference between them is to be equal to 2 pi by capital lambda. So it's almost similar equation with the compared to the fiber Bragg grating, but you have an additional negative is coming here. That is beta 1 minus beta 2 is equal to capital K here, but earlier it was beta 1 minus beta 2, because uh, the beta 2 is actually propagating backwards in the case of a fiber Bragg grating. So one interesting point to be noticed here is that since it is propagating in the forward direction, so while propagating in the forward direction, some of the energy is being coupled according to this equation. Some selectively, some wavelengths are going to be coupled into the cladding mode. So that is going to be absent in the core transmitted power. So if you monitor the transmitted optical power in a optical spectrum analyzer, you will find dips like this according to which cladding mode it is transferring power. Okay, but one thing to be noted here is that the periodicity requirement for long period gratings are not much stringent as in the case of a fiber Bragg grating. It is only 100 micrometer. This periodicity can be 100 micrometer, unlike the case with the FBG, where it was 500 nanometer. So it is very easy to fabricate this kind of long period gratings, and both the gratings are very much used in measuring various kind of parameters. So they are very good sensors. So how to create these kind of fibers? It can be, like the interference of UV beam. So you make use of a UV source and um, split into two, and then allow them to interfere in the core region of the or expose that interference pattern into the core of the photosensitive fiber. In that way, you can fabricate fiber Bragg gratings. Some of the important parameters of any kind of sensors is sensitivity. The sensitivity is the value of response per analyte concentration. I'm not generalizing this. For example, for a SPR based sensor that we discussed earlier, this can be a sensor for making a or sensing a chemical parameter or a biochemical molecule. So in that sense, the sensitivity, the value of the response per analyte concentration. Okay, how much it is, how the responsivity of the device, or my point is that the sensitivity should not be confused with how sensitive the device is. Sensitivity is something to do with the value of response per something that is actually detected. But how sensitive your sensor is can be, it is something related to the limit of the detection. Limit of detection. What is the minimum concentration, minimum value of the analyte that can be sensed? So the sensitivity, that is one. The detection accuracy, how accurate the measurement is. The limit of detection, what is the minimum concentration. So this concentration can be any other parameter as well. That's what I am saying. This is specific to this condition. You can change accordingly. The amount of analyte which can be sensed, selectivity or specificity, the chemicals interference. There can be other chemicals which interfere with this kind of detection, and that has to be minimized. Then response time, the time necessary for having about 95% of the response. Resolution, capability of the sensor to monitor the smallest difference in the analyte parameter. Okay, the resolution is not the characteristic of the sensor head, but it is the parameter that very much depending on the detector. Okay, resolution, the smallest difference that can be detected. Repeatability, how many times a sensor can give the similar result under similar experimental conditions. Reproducibility, the number of sensors manufactured, the same principle have similar response. So are you able to replicate, fabricate it again and again using the same principle? The noise, you are very much familiar. The random variation in the output signal, which has no information. Dynamic range between the limit of the detection and maximum concentration, which can be measured with confidence. So you have a minimum concentration in this particular case, minimum value and a maximum value. So that range is dynamic range. Linearity, a good sensor is expected to have linear calibration curve. It is linear when the output signal is directly proportional to analyte concentration throughout the range of the sensor. Drift, a small undesirable change in the characteristic curve with time. Reusability, can the sensor be used again and again? We have seen different types of fiber sensors and other sensors, SPR based sensors, etc. Can it be used again and again? In an economic point of view, that has to be used again and again, and if you are to buy it from a vendor, it's going to be very costly. Shelf life, what happens to the performance when kept for a long time? If kept unused for a long time, how it is going to affect the performance? The figure of merit, the ratio of the sensitivity of the sensor and the width of the spectral curve, which is something very specific to this kind of sensor we are talking about. But there are 14 to 15 such parameters, performance parameters for any sensor, any optical sensor that is to be studied. We have studied about microcavity sensors in detail in the last session, but I will simply mention one of the applications of such sensors in biology. So in biology, it is very common to use luminescent probes like fluorescent materials to label different parts of the cell or biological samples. Unless you label such biosamples with different fluorescing materials, it will look like transparent. So no color and too little contrast. There won't be much contrast between different locations in the sample. So that is the reason for this luminescent probes are used in biology. And we have seen what is a laser and what is a amplified spontaneous emission. But normally when you excite some material, it actually gives out fluorescent signal, which is spontaneous emission, which happens in all the directions. But a laser light is coherent. So it is always better to use laser instead of incoherent fluorescent light. for detection in order to improve the signal to noise ratio, especially while imaging the biological samples, etc. It is always desirable to have stimulated emission based experiments, even though it is much costly and complex. So this is one experiment which makes use of this biological sample, like a expressing cell. A green fluorescent protein expressing cell is kept in between two mirrors, and it is excited, and it is allowed to act as a laser. That means it acts as a microcavity, and it gives out its own laser radiation. So whatever changes that is happening in between, like the strain, pressure, etc., given to this cell biological sample, will be that signature will be there in the spectrum that is coming out of the laser. So it is a fluorescent material doped cell that is actually acting as a laser. So in this particular example, different types of micro cavities are incorporated in the biological cell uh, to monitor various dynamic fluctuations happening in the cell. So for this purpose, different micro beads or liquids are injected into the cell. For example, this is a cell, and this oil droplet is uh, injected into the cell, and this acts as a microcavity, and the corresponding spectrum is monitored, and this is actually the laser that is coming out. The frequencies can be thoroughly monitored while applying pressure, temperature, whatever external perturbations happening. So everything is monitored using the laser that is coming out, and the calculation for that. And this is adipocyte laser. For example, this is I'm coming to my conclusion. Before that, I will discuss this two final slides. For example, these are natural cells which contain lipids, which are called the adipocyte lasers. So adipocyte tissues, they have cells which naturally contain lipid droplets. The lipid droplet, we have already studied about the droplet laser. A droplet can act as a laser. So this droplet is naturally contained in the cell, and the one particular dye is used for making this material fluorescent, and the corresponding laser frequencies are analyzed.
Sorry to interrupt you, sir. The scheduled time is till 1:15.
1:15. Okay. Sorry. I thought it is going to be all by 1. Okay.
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 Yeah. Okay, sir.
Okay. So I was moving very fast. So similarly, these are polystyrene beads, that is polymer beads, plastic beads. Plastic beads. This is a plastic bead with a coating over it, and this is just a plastic bead which is injected into some kind of biological cell also, and the outside medium of the bead contains the gain medium. So here you can see the bead itself act as a gain medium, because the bead, the plastic sphere itself contains some gain medium, some fluorescing material. So in any case, this is a undoped uh, bead, plastic bead. This is a doped bead, and this is a undoped bead with a covering with the gain medium. So in all these cases, it can be very well go into the cell. Okay, these cells, this cell is called HeLa cells. This is a very important cells in biology. Somebody who is working in biology may identify this kind of cells, very important in cancer research, and they can readily internalize these beads for up to 20 micrometer. So if this is a cell, you keep this spheres, keep the sphere very close to the cell, and the cell internalize automatically, which is by a process called um, uh, this is a automatic process, endocytosis. The process is called endocytosis. By the process endocytosis, if such a bead is present here, the 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 cell will engulf that bead, and it will be inside the cell after some time. So that process is called endocytosis. So the cells can, by endocytosis, internalize such microcavities, and when we shine light, it can act as a laser, and these laser frequencies can be monitored from outside, and in that way you can label different number of cells using different sized spheres, microspheres, and spheres which are doped with different kinds of um, fluorescent materials. That means the fluorescent material, if one is emitting in the green, the other one can be emitting in the blue or in the orange. So it can be distinguished in terms of color or in terms of the size, because the size is going to determine the frequencies that are given out. This is such an experiment which makes use of this kind of beads, that doped beads inside the cell, and but the only difference is that you have multiple cells. Each of the cells either can internalize multiple number of spheres, but different beads different cells are internalizing beads of different size and different color. So when shines light on such laser, you can such a cell biological sample, you can see the laser that is emitting something like the whispering gallery mode lasing emission that is happening from that bead. And this intensity pattern or the image is analyzed using uh, hyperspectral imaging technique. Hyperspectral imaging technique can very well resolve the frequency components, the color components in this. And by clearly identifying the color of this ring, it can identify the size of the bead, because the size of the bead will determine the frequencies that are emitted from the laser. So you can see this is the original sample, and this is the hyperspectral image of that, and different beads are internalized inside different cells, and their positions are clearly located. So the hyperspectral imaging can further resolve into more number of cells in terms of the size of the beads and the color of the beads. And it is said that a large number of beads can be used to tag individual cells using different size and colors. So if you do that, the different fluorescence dyes can be used with non-overlapping spectra. So if you imagine one spectrum is not overlapping with the other spectrum, because different organic media, organic gain medium are used, and different size of the beads are also used. For example, 8 to the size range 8 to 12 micrometer. This is the range of the spheres, and a bin size of 2 nanometer. So with a gap of 2 nanometer, you can use around 2000 individual beads with different sizes, and you can use multiple colors, multiple fluorescent dyes, so that it will not overlap. So in that case, with 3 beads per cell, suppose you imagine that 3 beads are engulfed by a single cell by the process of endocytosis, and 5 different colored fluorescent dyes are used as gain medium in the beads. So that means it will be possible to tag around 10 raised to 11 cells. It is very much possible to tag uh, cells of this order, that is 10 raised to 11 cell, which is comparable to the number of cells in the human body itself. So this is a very interesting aspect of this WGM kind of sensing and lasing. So these microcavities find huge applications in biology, and in all other kind of sensing. I think I will stop here for discussion. Thank you.
Now let's move on to the questionnaire session. If you have any doubts, you may ask now by posting them in the chat box. There are so many questions in the chat box. I will ask the questions. The first question is asked by Mr.
I can
Mukesh
see
Kumar.
that. So I can see.
Yeah.
Okay,
Yes, sir.
you can tell. Okay, you can tell.
Yeah,
Okay.
I will tell. Yeah. The first question is asked by Mukesh Kumar. The question is, can microcavity be used for Q switching, cavity dumping, and mode locking to generate ultra short laser pulses? Because microcavity might reduce laser intensity appreciably.
So Mukesh Kumar. Huh?
Yeah, Mukesh Kumar.
Okay. Mukesh Kumar, we are talking about a different scale. Obviously, if you are talking about very small, ultra small lasers, which is to be Q switched, maybe this may be useful, but these are very small in their structure. Okay, so you can use it if your laser is of this size. I am not very much familiar with that kind of application. I have not worked with it, but maybe possible.
I think your doubt is cleared, Mr. Mukesh Kumar. Another question is again asked by Mukesh Kumar. The question is, what is gallery modes?
What is gallery modes? Okay. So I showed you a figure, Whispering Gallery, St. Paul's Cathedral, etc. Remember that? So this is a gallery for acoustic resonance. So here the acoustic resonance is happening. So this is a gallery. The people can whisper from here. It will be clearly audible on the other side. So the losses are very minimum. So why it is minimum? It can propagate because the resonance is happening there. So that was for acoustic waves. But similarly, the optical waves can be resonated. They can be very well contained in a structure like this, any spherical, circular, dielectric structure. So from the same origin, the name has the same origin as the Whispering Gallery available in the St. Paul's Cathedral. So this is, we are talking about the light, but here it is sound.
I hope your doubt is cleared, Mr. Mukesh. There is another question asked by Dharanya C. The question is, how plasmon excitation happen? What is the difference between radiative and non-radiative SPR?
As I mentioned earlier, the you have to have a interface between a metal layer and a dielectric. Okay, and also the momentum has to be conserved. So depending on the direction and the angle of excitation of your laser pulse or your source, there can be a collective oscillation of the electrons happening. So it cannot happen all the time. Okay, so in the Kreshman configuration, the angle can be clearly calculated once the dielectric constant values of the metal and the dielectric is known. And usually in the dielectric surrounding, you have other materials, analytes present. So the presence of analyte is actually being monitored by the uh, SPR, that is from the dip in the transmission spectrum. And also there are other calculations are also there, not just from the dip. It makes use of many algorithms to get more details about the process and the analytes.
Okay. The next question is how plasmon resonance frequency depend on refractive index?
Okay, that theory I'm not displayed here, but as I mentioned here, the in order to conserve the momentum at a particular angle, what is important is the surrounding dielectric constant. It's a combination of the analytes, or as we mentioned earlier, there is a region, there is a depth up to which the evanescent wave can interact. So maybe beyond 100 nanometer or 50 nanometer, the evanescent field is almost zero. So no interactions of electric field with the medium. So within that region, there can whatever things are happening can be clearly monitored. It can be imaging, or it can be what you call some antigen antibody kind of binding. So that are monitored. So can you repeat the question once more?
The question
Hello.
is how plasmon resonance frequency depend on refractive index?
Yes, refractive index means the electric constant. Yes, but I am unable to do the entire theory here. But the, what it depends is on the surrounding refractive index and the type of metal film that you have coated, and the angle at which uh, the the radiation excites uh, the plasmon.
I hope
And
your
of
doubt
course,
is clear.
this is a traveling
Yep.
kind of plasmon that is generated on the film, unlike in the case of a nanoparticle. So okay. I'm obviously I'm not doing any no experiments on this. I have no any practical. I don't have any practical experience on these kind of experiments, but whatever thing I know, I'm just sharing with you. Okay.
Okay, sir. I hope your doubt is clear, Mrs. Dharanya C. The next question is uh, does the dark current affect the resolution of sensors?
Dark current. So you may be coming from the electronic background, right? Hmm? So the resolution is not defined in terms of the device as such, but it is defined in terms of the detector, the type of detector. So you can, for the same setup, you will be making use of types of detectors. So if the detector is different, it's so the dark current values may be different. So obviously, the resolution can affect. That's what I understand.
So the next question is microcavity stable cavity or unstable cavity?
Mm, cavity, I don't know how to categorize it, just like any other laser cavity in a commercial laser. But what is important here is, just we can make the laser or the light to be there for a longer period of time. So that Q value, the quality factor is very high in the microcavity. So we can contain the light for a longer duration of time. I don't know the exact classification, whether it is stable or unstable, depending on the shape, because the shape is also very important. There are different types of microcavities based on spheres, based on cylinders, then ring structure, toroids, a lot of things are there. So I don't know whether it can be just classified certain number of microsphere cavities as stable, the other ones unstable.
So the last question of the session. The question is how higher order transverse mode in microcavity are filtered out to lace only in fundamental mode of laser.
So I have done one experiment. I mentioned one experiment which made use of a hollow fiber which is coated with a gain medium inside. Micro ring embedded. That's what I did. So there are a lot of modes. There are a lot of modes available initially, but when we increase the pump power, the side modes were suppressed. And this is because the normal WGM modes are not applicable here, but the WGM is actually converted to two modified WGMs. If you remember, you can go through that paper, and we have referred two such papers which were published earlier to come to that conclusion. So the WGM is actually modified, and the new two sets of modes which are called the modified WGMs, they interfere constructively only at certain conditions. So the conditions become stringent, and the modes selection was done, and we could find a single mode operation.
I think it is time to wind up the session. I take this opportunity to thank Dr. Kailasnath M. Thank you, sir, for the excellent presentation. Your years of research, your ability to present the subject in such an interesting manner is really appreciable. I would like to thank you on behalf of all the members present here. I hope you all enjoyed the presentation. Thank you so much for your interest and attention. The next session will be at 2:15, and I hope all of you will join at sharp time. And this brings us to the end of the second session. Thank you all.